Okay, it looks like most people are here. Are you ready, Anthony?
Yeah.
All right. Well, let me get started. If everybody can see with your screen up, but I wanted to. Maybe Anthony, maybe stop sharing your screen for just a minute.
Sure. I think that worked.
Better, so everybody can see each other.
Hey, Dave, your volume's kind of low. Is it possible you have the wrong speaker microphone?
Maybe it's just I need a new voice. How's that? Is that better?
Not really, but that's okay.
I'm not going to speak for very long. I just wanted to say a couple of things. One is I wanted to start with the metaphor where the road to the highest academic degree that we have is a long and winding road and complicated by many things, and so in many ways it's like a flower. So I wanted to use that as a metaphor for sort of celebrating all the things that that has allowed Anthony to get to the point where he is currently. And just a few comments. I mean, Anthony, interestingly enough, started with a passion for photosynthesis, probably the most important process on this, for our biosphere, at least on this planet. And he started at Emory right after a student, Sha Li, had discovered the notion of the energetic constraints on making peptide bilayers. Peptide bilayers are like lipid bilayer membranes, which most of us know, but composed of peptides. In this case, the limiting factor on the assembly of the peptide bilayers is actually the leaflet interface that ties them together. And she explored this in the context of making mixed charged peptides, positive charged, internal positively charged, and internally negatively charged side chains in such a way that the interface then was electronically neutral. But what that did is that association was so strong that it created an incredible electrochemical potential across the membrane, because that meant that the one surface of the membrane was all positively charged and one surface of the membrane was all negatively charged. So Anthony, recognizing the power of being able to control the assemblies through mediating the leaflet interface, decided that he could do that with metals. And that's what he's going to talk about, his description of the structure of these bilayers that are held together or nucleated or templated by the presence of metals. The kind of functional capability that gives these assemblies and makes them possible. It's interesting that what he's ended up with is not all the way to photosynthesis, but it's still able to interconvert chemical structures by using a large array of metals that can control a large redox transfer from the metal assemblies of one redox state to substrates of another, which is what he'll talk about. I will say one final thing, and that is 2020 is probably not the best year to finish one's PhD. And Anthony has had the fortunate opportunity to really lean on and depend on many of you that are here to help complete some of the experiments that he needed, to help in some of the analysis, and maybe just to keep him sane. So I wanted to first of all thank you all for the contributions that you've made in this crazy year that we're in to Anthony. And I also wanted to celebrate Anthony's ability to coordinate all that. So with that, the stage is yours, Anthony.
All right, thank you so much. Let's see if I can get this shared again. Okay. Okay, so do we all see, a slow dance of molecules on your screens?
Yes.
Cool, perfect. Okay, well, yes. Again, Dave, thank you for the lovely introduction. And I also want to thank everyone for joining. I know Thursday afternoons tend to be busy for most people, so I'm just really grateful that you're able to permit me and indulge me the hour to discuss my work here. And in a nutshell, I've seen how short peptides can be engineered to build themselves into intricate networks that bind metals like copper, as seen in this movie over here. And I discovered that these networks bind activate these metals. Awesome. Now,
Anthony,
yes.
you just blipped out, at least for me.
Can we
Yeah,
all,
close all your Chrome tabs again.
they're all already closed. Well, that's Zoom for you. The only applications I have running is Zoom and PowerPoint.
You're good right now, so talk fast.
Okay. Let's see if there's anything else I can do to speed up bandwidth. Nope. Okay. Well, we're just gonna run with this, but leave a comment in the chat or something, or just call out again, and I can repeat anything that might have got glitched out, definitely. But yes, so before I'd like to dive in, here's just a brief outline of today's talk. I'm just going to first introduce the background that inspired my research. I won't be covering everything, but I'll be devoting about 15 minutes to the following two projects. The first is concerning how I resolve the structure and electrochemistry of these copper peptide networks. And then the other project is probing how changing the oxidation state of that copper ion reveals what I call valence-dependent effects on the formation and reactivity of these structures. And there's another project where I resolved these transient structures that form en route to these more intricate arrays. But I already published that information, so if you're curious, I encourage you to read the article, and you can certainly follow up with me after the fact if you want to talk about it, or if you need help getting through the paywall, I can send you the PDF. But without much further ado, I'll start off as sort of Dave alluded about photosynthesis, which is, in my opinion, the coolest feat of chemistry in the universe. I know that's a big claim, but this diagram over here briefly summarizes how biology generates a wireless current that stems from light and water, and it ends in food. And it does all of this with 100% quantum efficiency. It's actually so perfect that biology creates enzymes that leach the power from these electron transfer chains, and it winds up dumping it into dissolved oxygen or water, and it's a huge waste. But without those safety valves, the power you generate and bleed out from photosynthesis would actually fry the cell. So that's cool, but why is it relevant? Well, starting out, my goal was actually to synthesize a structure that would act like an electron bank, basically a network of biomolecules that could build themselves and carry all these electrons should a cell run a surplus. And for inspiration, I'd like to take a look at how biology does it. Electron carriers like plasticinin, which I've circled here in red, these are proteins that bind one divalent copper cofactor, which I've shown over here. And this is what ends up relaying electrons from cytochrome B6F to photosystem 1. So plasticinin on its own has this innate electron storage capability. It just does it one at a time. And so this is what inspired my research question. Can we build a super plasticinin? Something that is, proteinaceous in nature, but it could bind not one, but hopefully thousands of copper ions and retain its fold and its function, no matter how much you charge it up. And what we're essentially trying to do here is increase something called, the active site density or the active site ratio. And for reference, plasticinin has one copper cofactor, so one electron storage potential, for every 11,000 Daltons of mass. And we're going to try to beat that by the end of the talk, or maybe even sooner than the end. And it's a big goal, but I hope you'll find that it's reasonable. But before really sinking our teeth in, I just want to show that there is precedence for tapping into this wasted current of photosynthesis. There are some researchers in 2016 that were able to engineer rat cytochromes into the chain, and those were able to siphon off electrons and dump them into atrazine, which is an herbicide, photosynthesis poison. And that's certainly useful, but I wouldn't consider that storing the electrons. You're just burning it in a slightly more useful way. All right, well, where do we begin with this ambition of electron storage? I hypothesize that this short peptide called K16A, it has all the ingredients for superplasticinin already. It has two domains I'd like to highlight. The first being this N-terminal histidine dyad, and this is what binds metals really well. And if you have sharp eyes, you might have seen that plasticinin in that previous slide has two histidines binding copper as well. But this compound is actually 10 times smaller than plasticinin, and it's 1,000 times more soluble. But when you dissolve it, it doesn't just, disperse. It's actually going to self-organize thanks to this nucleating core sequence that I've colored in red. And these amino acids like to stick together and drive these peptides to assemble into intricate nanofibers and nanotubes even sometimes. And you get those different structures depending on what metals you throw at the histidine dyad. So early on, the methodology was, come to lab, grab a dart, throw it at the periodic table, and you dissolve K16A with whatever metal struck bullseye that day. And yeah, a good number of those metal ions we surveyed produced, just junk, frankly, amorphous aggregate. Or you would get fibrils one day, but then junk the next day. And I'm highlighting those cases with red and yellow stars, respectively. But we did get a few hits on, thankfully, the cheaper metals like nickel, zinc, silver. That was actually a little bit unexpected. And also silver's first row cousin, copper. So anyway, these are, in green, those are the reliable structure makers or metal templates. And whoops, sorry. Let's go back. What I want to highlight is that first in our lab, we studied zinc actually. And zinc is cool because when you co-assemble zinc with K16A, at first you get these banana crow-like structures. And what's interesting is as you ramp up the zinc concentration, these banana curls are going to anneal into continuous peptide nanotubes, which was pretty alarming in a good way. But copper is also cool, and that's because regardless of the copper to peptide ratio, we see these elongated nanoribbon structures form under the electron microscope. And that I thought was a sign of robustness. And also copper is more reactive than zinc, and that's what plasticinin uses. So copper is ultimately what I ran with. And generally, there are two ways that you can metallate K16A. First being you can pre-assemble the K16A peptide into these nanofibers, which I'm showing in this EM labeled A, and here's a cartoon rendering of it, also called A. And remember that red segment is going to build itself up over time on its own. So we don't need metals to grow it necessarily. So letting it grow and pre-assemble beforehand will give you these fibers. And then after you get these mature fibers, you can add a concentrated copper fluoride solution, and that's going to result in the slightly thicker fibers, which I'm showing in panel B. And what we suspect is happening here is that the copper is bundling the fibers by way of these histidine-rich surfaces that extend along the structure. And we've characterized this process before with several other divalent ions. So that's pretty cool. On the flip side, you could take the peptide powder and you dissolve it and then immediately add the copper solution. So before you get a chance to get these fibers, and what happens instead is you get these twisted nanoribbon structures like I've shown on the previous slide. And I thought that was kind of interesting on a fundamental level, and I had this inkling that these structural gross anatomical differences were stemming from differences in coordination. So one method for estimating a metal ion's ligand composition is through its d orbital transitions. And here I'm showing the electronic spectra for co-assembled K16A in blue, and pre-assembled K16A is in green. And just to remind you, these are the nanoribbons, and these are those bundled fibrils. And as a control in red in the inset is just hexaaquacopper, which is copper with no peptide. And yeah, ligation by the peptide indeed induces a drastic blue shift from the near IR to 573 nanometers for the co-assembled case. For the pre-assembled fibers, that blue shifts not as much, but still significantly to 600 nanometers. And these differences, they would indicate that there are structural distinctions between both preparation modes. But it's difficult to say exactly how they're different just looking at lambda max. That was at least until I came across this equation. And this equation over here can approximately relate absorption to ligand composition in only square planar copper coordination complexes based off of lambda max. And according to some quick calculations that I ran, it would turn out that complex with 2 imidazoles and 2 amino groups, which is to say 2 histidines and 2 N-termini, that is going to be most consistent with this co-assembled K16A ribbon structure. And that's the structure that I'm actually most interested in. And this slide describes why. Cyclic voltammetry on these co-assembled ribbons will yield a consistent wave shape across all cycles. And we're assigning these broad symmetric wave fronts to the copper 1, copper 2 couple. And they're undergoing slow redox kinetics with a rather high reduction potential of 670 millivolts versus a standard hydrogen electrode. However, when you compare it to the bundled fibrils, there's really no discernible redox couple. And that's also the case for the unassembled particle phase of these copper ribbons. And just to explain more what's going on here, plots A and C are the same sample, but this didn't have time to mature into these elongated structures. And so this tells me that the ribbons co-assembled nanoribbon morphology, that's essential for obtaining this reversible redox property. And that's important for an electron bank. You know, a bank where you only make deposits but no withdrawals is probably better described as a Ponzi scheme. But anyway, a Randall-Sebchik workup on plot A, which I've shown over here in D, this shows that the scan rate actually correlates quite linearly with both anodic and cathodic currents. And this kind of relationship is really only seen with fast diffusing systems. But when I calculated the diffusion coefficient for these giant assemblies, that came out to 10 to the minus 13 centimeters squared per second. And for reference, plasticyanine, which I love, is 10 to the minus 7. So these giant, you know, nanostructures, they're 1,000,000 times slower than slow. That would give us a bit of a paradox at first glance. These ribbons are bigger than plasticyanine and they're less diffusive, but this linear Cottrell-like relationship is frankly on par with just hexaaquacopper, no peptide. Typically metalloproteins, you don't even get to make these plots because of issues with diffusion. But it turns out, if you look at the literature, that the combination of quasi-reversible but slow diffusing analyte is an indication for charge hopping. And that means that the electrons there that we're pumping into and out the assembly through the apparatus, that they're literally hopping across multiple copper sites to circumvent the limits of deposition. So now I'm really curious about copper K16A structure. But in order to deduce how K16A is binding copper, we're gonna have to resolve the overall peptide scaffold first. And to save time, it kills me to do this, but I have to skip over some details. But in short, we resolved a typical cross beta architecture using powder X-ray diffraction, atomic force microscopy, and solid state NMR. That latter technique in particular deserves hours of discussion. It is incredibly fascinating, but altogether, I just have to zoom to the highlights and reveal that the peptide network exists as a bilayered stack of beta sheets. And the peptides alternate anti-parallel down the hydrogen bonding axis. Also, these peptides, we call them doubly out of register, which means the alternate facing histidine dyads are skewed outwards to the extremities and they overhang. And that's actually pretty significant because when these histidines overhang, they're able to interlace at the bilayer seam. And in the model that I moved over there, where I've rendered all the histidines in blue, you can actually appreciate, hopefully, that the site of greatest histidine density is where the two bilayers meet. And that is where we infer where most of the copper peptide interactions are occurring. So that's where we have to sort of zoom in on. This is going to be crucial knowledge in understanding the next steps in resolving the coordination sphere. So I know that last slide was probably a bit of a whirlwind, and I just want to offer another view of the model as we have it defined so far, where we have these two stacks of beta sheets that extend lengthwise through the hydrogen bonding interactions into the plane of the monitors, and the zone of greatest histidine density and N-terminus density, that is again where these two stacks come together. And we confer the width of the nanoribbons by stacking of these beta sheets. And this is typical amyloid, quaternary structure. So we actually know quite a bit about the model so far, but as that glowing blue bubble of warning told us, we still have a few unanswered questions, like how many histidines or N-termini or waters or who knows what is actually participating in copper chelation, and what's their geometries and orientation. So to address this, we use electron paramagnetic resonance through our collaboration with Kurt Warnke's lab. And in this experiment, we show the EPR line shapes of copper K16A with varying ratios of metal. And each of these lines corresponds to a different preparation where I've increased the metal saturation from 10% metal to like 80% metal. And the key takeaway here, well, there's a few key takeaways. The first being these multiplets that happen around 320, 330 millitesla. And we notice that these are best resolved between 0.1 and 0.2 equivalents of copper. The number of these so-called super hyperfine features is consistent with a copper that's bound to 2 or more nitrogen ligands. And that could be either the histidine or the N-terminal mean or 1 and 1, or more than that. So that's a significant step closer. Additionally, we see these features smooth out as we increase the metal content. And this is interesting because it suggests the appearance of more chemically distinct coordination geometries that result in signal broadening. So the way the copper K16A binds copper is actually changing with metal ratio. And this surprised me because if you recall from one of the earlier slides, the gross morphology of those nanoribbons does not appear to be sensitive to the metal peptide ratio. But if you zoom in to the coordination sphere at the angstrom level, well, that's a different story. And last thing about these data, at 0.8 equivalents, we see a G parallel resonance appear at 260 millitesla, very slight. And this is consistent with hexa-aqua copper. So what this means is that when we approach 0.8 equivalents of copper, we've actually saturated the complex and it can't hold any more metal ions. So that tells us, you know, our deposit limit for our electron bank, so to speak. Now, altogether, EPR has given us, I think, quite the head start here. And that, again, that structural heterogeneity that we've inferred is important because for the higher resolution techniques that are coming up, we deliberately prepared samples at 0.2 metal equivalents so we could best isolate our most resolvable structures. Okay, next I performed a technique called X-ray absorption fine structure, XFs for short. In a nutshell, it's a way of identifying a metal's ligand composition and geometry. And it's pretty technical, so I'll explain it as we go. But on the left, the black curve is essentially the absorption spectrum for copper's core electrons in copper K16A. And at certain X-ray energies, these electrons are photoexcited into the continuum where they propagate from the copper nucleus as a spherical wave. But above copper's K edge, the absorptivity fluctuates. And that's because that spherical electron wave is scattering against nearby atoms and scattering then back to the copper, where all sorts of quantum mechanical sorcery happens. And that results in these very peculiar absorbance oscillations. You change the ligands, you change their positions, and you're going to change the ripple patterns. Specifically, there are interferences at 9,030 and 9,050 electron volts, and these are diagnostic for histidine coordination. So this tells us that for sure the histidine dyad is interacting. It's not just the N-termini. Another hallmark feature is the smoothness of the K edge. And this smoothness is textbook for a square planar geometry, which was an assumption up until now, because there's a transition at the pre-edge associated with a 1s to 4p excitation that's forbidden in centrosymmetric geometries. And that transition becomes allowed, though, when you reduce the copper 2 to copper 1 through the addition of a reductant like ascorbate, which is vitamin C. And we can see that experiment in the red plot, and I've zoomed in on the pre-edge zone in this inset. And you can see that concomitant with reduction, we achieve this structural rearrangement from a square plane to a tetrahedron. And since tetrahedrons don't have inversion symmetry, selection rules don't apply, and the 1s to 4p transitions become allowed. Long story short, that's evidence that copper K16A has a square planar geometry when it's oxidized. And after doing some arithmetic to these post-edge oscillations, we can get even more information from the data because we'll extract out this convolution of scattering functions that we can then process further into something called R space. And fits to R space for copper K16A will inform that we have a combination of 4 nitrogens and or oxygen atoms binding a common copper center with bond lengths of 2 angstroms each. And that we have anywhere between 1 to 3 histidines binding, which is admittedly a range, but we can narrow that down further with another experiment coming up. But I want to just quickly recap that XFs has provided us several structural parameters to work with, like a definitive coordination number, date of bond lengths, and the knowledge that redox is able to flexibly change or rearrange the coordination structure quite drastically. And that's not guaranteed in giant, delicate networks like I'm studying. So that was quite the boon. Okay, the next experiment is electron spin echo envelope modulation, and ECM for short. And this experiment was made possible again by our wonderful collaborators in Kurt's lab. And like eSeam, or like EPR, eSeam is measuring the chemical environment of the unpaired electron on copper-2. And when you spin excite that electron with a radio frequency pulse, you'll yield coupling interactions between the copper and the distal nitrogen on the imidazole ring, or the tau nitrogen. So that makes eSeam a really good technique for resolving these elusive histidine ligands. Conceptually, the workup actually resembles XFs to a degree. The signal we obtain is a complicated waveform, and a Fourier transform can be used to convert the time domain to a frequency domain. And here's a quick way that we can fingerprint the data. If we look for what's called double quantum combination lines, we can identify this resonance, 1 VDQ, so that means 1 histidine, and much weaker but still measurable is 2 VDQ, and that's 2 histidines. And over here, there's no 3 VDQ, so the buck stops there. Altogether, this identifies that we have a double histidine contributor as a structural component to copper 2K16A. And if you thought counting VDQs was a little boring, you could always do a 15 parameter fit to the line shape and get a whole bunch of extra structural information from the data, such as the dihedral between both histidine planes. And what we can tell from the Euler angles in the fits is that the histidines are canted to each other by about 94 degrees. So they're almost like this, perpendicular to each other. And if you were to then look at other fitting parameters, you would next find that the angle between the sensitive nitrogens with respect to the copper ion measures out to about 140 degrees. Now, it's kind of hard to visualize this in a simplified 2D cartoon as I've shown it, but that 140 degree angle doesn't exactly apply to the angle of the chelating nitrogens, which is called the bite angle. And that's what we really need to resolve the coordination sphere. Because depending on side chain positioning and all sorts of parameters, many bite angles can accommodate this 140 degree angle criterion. But we still have, I want to highlight, 2 additional spatial restraints that complement the XF's data. And my next goal was to merge these 2 experiments to have a more informed and comprehensive model. And that's where the molecular dynamics comes in. Taking in the NMR, the AFM, and powder XRD data, I constructed this peptide lattice in silico. And once that equilibrated, I placed a dummy copper atom in the middle, and I restrained it to the nearest histidine pair with bond distances taken from XFs and the histidine angles and dihedrals I took from eSeam. And that resulted in this lovely little movie that you saw earlier. Okay, good. Now, besides admiring it all day long, we can actually extract some quantitative information from this. A cluster analysis reveals that the force fields respected all experimental restraints, and they yield a conformation distribution that's consistent with the data. Bond distances averaged out to 2 angstroms, and the imidazole dihedral, which I'm showing here, maintains at 94 degrees. And what's more, this was able to average out and give us an average coordination sphere, which finally lets us infer that coveted bite angle. And that averaged to 90 degrees, while still maintaining the 140 degree criterion taken from eSeam. So what this affirms to us is that the structure is a cis-bis-his geometry. And that just means cis means that the ligands are 90 degrees from each other, as opposed to trans 180. And bis, I think, is like Latin for 2. All right, now that was pretty cool. I really like molecular dynamics. The movies, I think, are really interesting. but the same can be said about Star Wars, frankly. And we all know how scientific those movies are, so we have to convince ourselves that the simulations reflect reality. And what I did to do that is I took the coordinates from the final frame and I used a program to compute a diffraction pattern from it. And the result is in this plot, and we can see that there are two reflections that resemble the hallmark amyloid D spacings of about 4.7 angstroms and about 10 angstroms. And this corresponds to the hydrogen bonding distance and the lamination distance between the beta sheets. And these match the experimental data that I showed briefly on an earlier slide. So this convinces me that the computation is reasonable because, after all, this shows the force field is respecting coordination sphere and the global quaternary structure for the system. But I also mentioned several slides ago that the electronic transitions suggest 2 histidines and 2 N-termini are binding the copper. I just showed histidines binding. So the next thing that I did was I applied the x-ass restraints to the N-termini as well, and that yielded a square planar structure, which I'm showing here. And this is even more consistent with the data that I've described, except still for one key difference. There's only one dummy copper in here, partly because including these dummy coppers is annoying, but also I wanted to observe this in isolation. However, if I'm making claims that the peptide lattice is concentrating the metal ions to facilitate charge hopping, it would be nice to estimate the range of possible copper-copper distances with these informed parameterized trajectories. To do this, I included 4 more dummies in the nearest available histidine pairs in each cardinal direction, and I extended the trajectory further. And this was the result. What I've done here is I've rotated the view 90 degrees about the y-axis so that bilayer interface is now coplanar with our monitors. And that will let us see that the average copper-copper distance ranges from 10 to 12 angstroms, depending on if you're going between adjacent laminates or you're measuring within a single laminate. Whoops. Let's go back. And okay, this movie, I think, now offers a time averaged vantage on how the peptide lattice is displaying these metals. And here I've hidden all the side chains except for the chelating nitrogen on histidine, and I've showed those as blue bubbles. And I've superimposed their positions over time. And those 5 dummy coppers, those are yellow bubbles in the middle. And I know it looks like there's 100 of them, but keep in mind, this is a snapshot of their positions across each frame of the movie. And that tells us that we have a distribution here. And as this ensemble swivels, take note of how the D spacings punctuates the blue bubbles into these distributed yet periodic regions of interest. And this in turn is anchoring the bound metal with a similar flexible periodicity. And that's what I'm arguing is the reason for charge hopping. That's what makes it possible. It's the combination of fluidity with, or paracrystallinity might be a better word, with this Goldilocks proximity. Okay, there we go. Okay, but naturally, the next question you may want to ask is, well, is a 10 to 13 angstrom copper-copper distance with flex, is that truly reasonable for charge hopping? And to get a numerical sense of that, we can refer to this equation known as the Dutton ruler. And this postulates that electron transfer in any protein actually is a function of the donor-acceptor distance, the reorganization energy, and the redox couple's thermodynamic driving force. And we can assume that delta G is 0 because the copper sites are all identical and site similar. For the reorganization parameter, well, that's actually, that has to be measured empirically, and it's non-trivial to do that. But a really common estimate used in the literature is 0.7 electron volts. And that's what I've used here. And for R, the copper-copper distance, we can plug in the upper and lower limits taken from the molecular dynamics simulation. And that output gives us an electron transfer rate constant of 2 times 10 to the 6 to 10 to the 7 inverse seconds. And that's a pretty decent rate constant, and it's well within normal biological electron transfer regimes. You know, and biologically, the upper limits for electron transfer has been measured out to hops of 14 to 20 angstroms. So we have room to spare here. Now, quickly circling back, I hope that I've convinced you that copper K16A can behave like an electron bank. And if you recall, the active site density for plastocyanin, that's 1 copper for 11,000 Daltons of mass. But for copper K16A, it's shy of 0.8 coppers or 2,000 Daltons of mass. And that gives us a roughly 5-fold improvement of over plastocyanin at identical concentrations. But consider that the solubility limit for most proteins is actually in the low micromolar range. However, when I do my experiments, I dissolve these peptides at several millimolar, actually. So really, the electron capacity for this system is more like a 5,000-fold improvement in reality. Now that we've considered copper K16A's structure and redox, this can now let me ask additional questions like, how do we get electrons in and out, and what are the consequences of doing so? To refresh, I'm going to circle back to our muse plastocyanin. If we zoom in over here, we can see that this interaction where plastocyanin sources its electrons by docking cytochrome B6F, that cytochrome B6F has several electron transfer relay sites contained within. And these electrons are in turn sourced from an upstream carrier pool called plastoquinone. And plastoquinone looks like this. It's a small molecule that can undergo reversible 1 and 2 electron redox. It's somewhat broadly recognized by the proteome because the Q pool is not only able to insert at multiple sites within cytochrome B6F, but it's able to interact with several other photosynthetic proteins, like the ones designed to bleed out photosynthesis. Now, I'm mentioning this because I'm hypothesizing next that small molecules like quinones are going to better oxidize and reduce copper K16A reversibly over something even larger and more delicate and specific like cytochrome B6F. So these are the next 2 research questions that will be the center for the final third of my talk. And that's, can we use a synthetic Q pool to facilitate reversible redox on copper K16A? And how will redox change its structure and reactivity? Okay, excuse me. Now, moving forward, the first thing I needed to prove to myself was that bulk copper K16A redox cycles, which isn't a guarantee, like I said, for these kinds of assembly systems. And I predicted that if we reduced copper 2 to copper 1 K16A, it would react and cycle that stored electron onto dissolved oxygen, and that would generate in turn hydrogen peroxide. And we can detect peroxide evolution using commercially available kits through fluorophores. Now, as a control, I first tested aqua copper, which again has no peptide in it. And on its own, after you reduce it, it over time will generate peroxide, as shown by this red line. I should note that I'm using the ascorbate as the reductant here instead of quinones. And that's just because at the time I knew for sure ascorbate worked because of the XAFS experiment. I promise the quinones are coming really soon. But anyway, the next sample that I tested was unassembled copper K16A, which is the black curve. And this is similar to, it's the same preparation that I showed for the cyclic voltammetry experiments. It's the same as the copper ribbons, but it never had time to mature and form structures. So since it's disordered, it seems to therefore have a similar turnover and rate compared to just hexaaqua copper. However, when we compare pre-assembled copper K16A, which is the green curve, and co-assembled copper K16A, which is the blue curve, their overall turnover decreases significantly. Well, why could that be? The way I view it is pre-assembled copper K16A is actually a monolayer, and the metals are sequestered externally by decorating the histidine-rich fibril surface. So half of those coppers are buffered away by peptide and protected from dissolved oxygen. And that makes it harder to reach and react. Even more so, the copper ribbon structure, those copper ions are sandwiched between two leaflets, and that makes them even more buffered from the reactant in the solution. And running again with that electron bank analogy, this is good because we want our assets protected in our electron bank, but still somewhat accessible. So just to quickly summarize, what we're kind of showing here is that reduction by a reductant occurs really quickly within mixing time, but reoxidation, getting those electrons out, that takes more time. Now, so those experiments with the hydrogen peroxide demonstrated turnover in mature copper 1 K16A, but now I'm wondering if reducing copper 2 to copper 1 will affect how it assembles with K16A. So to do that, I prepared two samples, one as just copper 2, and that's shown in these top pictures, and the other, I reduced it to copper 1, again, using ascorbate, and I left that in an oxygen-free glove bag. So no oxygen was robbing the electron banks, so to speak. And something happens. In the top row, we have a vial and my lovely fingers as showing just the copper K16A solution, and it's just a thin pale blue solution. You hold it upside down, it just flows to the bottom, nothing exciting. But when you reduce it to copper 1, something interesting happens because you get, instead of a pale blue solution, we get this congealed translucent gel that will actually stick to the bottom of the tube when you invert it. So something here has changed, and electron microscopy indeed reveals anatomical differences. If you recall, copper 2 K16A forms nothing exciting at first when you mix them, but over time, it's going to mature into these short, fat, twisted nanoribbons. And even if you reduce the bound copper over here, after it's mature, you're not going to change these structures. However, if you take the freshly prepared copper 2 K16A solution and reduce it, you immediately get some structures propagating, and those mature into more elongated and slender fibrils. And it also makes more of them. Just to be able to resolve these individual structures, I had to dilute the aliquot tenfold. That's how much we obtained, and that explains the gel consistency. This copper 1 K16A, it also makes the fibrils really fast compared to the ribbon structures. Okay, how fast are we talking? Well, circular dichroism can figure that out for us. For copper 2 K16A, the ribbons, we see an ellipticity minima that reads initially at 192 nanometers, and that's consistent with a random coil. And that backbone is going to remain disordered until the sigmoidal region around day 40. And that's where the ellipticity minimum begins redshifting towards 212. And that signal is consistent with pi to pi star backbone excitations that's found in beta sheaves. So we consider the structure to have matured once it reaches this, once it plateaus at this redshifted value. Copper 2 K16A, therefore, takes about 70 to 80 days to finish growing. For copper 1 K16A, which I've shown in this plot in black, with one equivalent of ascorbate, we can see that it too has an initial ellipticity minimum at 195 nanometers, and that redshifts to the mid 210s. But take note of the x-axis here. We max out at 90 minutes. If you say that another way, copper 2 K16A is going to take 2 months to grow. Copper 1 takes 2 hours tops. Even if you have the amount of ascorbate, which is the red curve, that slows the growth, but only by a rough factor of 2. So this is pretty interesting. And, you know, I'm not a patient person. And copper 1 K16A is looking more attractive by the minute, because not only does it make more assemblies, but it does so 500 times faster, and it preloads them with electrons for us.
Can I ask a quick question there, though?
Of course.
So you're talking the time scale of the assembly for the copper 1 system is much shorter, as you show, but it's not assembling the same structure. Is that correct?
Yes and no. That's a little tricky to pin down, because if you look at it, you know, from a bird's eye view, they don't look too similar. One's really long and thin, the other's short and fat, and they has more lamination interactions. But comparing the atomic force microscopy data, they're both bilayers. Their electronic transitions are both 570 nanometers within a range of plus or minus 3 nanometers. What else?
But they are different structures, because like on the previous slide, you said that they were slender fibers, right, for the copper 1 growth. And for the copper 2 growth, it's more laminated, as you said, I believe.
And so what I think, I can show you this after I'm done. I have some XRD data comparing copper 1 and copper 2. They have the same D spacings, but the relative intensities for the hydrogen bonding reflection and the lamination reflection, they correspond to the aspect ratio. So I think like the best analogy I can think of is, you know, maybe like a person that's really tall and skinny versus someone that might be overweight and short. I hate to like use those kind of terms, but you know, it's like they're the same.
Very un-PC, Anthony. Very un-PC.
Excuse me.
No.
The same species, you know, but it's just the direction of growth is not identical. That was the first analogy I can think of off the cuff. Forgive me.
but it's very important. So some of what you're talking about structurally here is copper, it seems, or the ligation sites being similar in the two assembled structures, but the assembled structures themselves are quite different.
From the grossly, yes, but at an atomistic scale, I don't know.
But at an atomistic scale from a very singular perspective, which is the copper sites, right?
I mean, that's a bit of a toss-up too, because the electronic transitions are identical within error. And
electronic transitions of what?
Of the copper center.
Yeah, so that's the copper center, but that's a very local region of these structures. It's not surprising that copper would have a similar ligation field in different structures, right? I mean,
that is possible.
It has a certain possibilities available to it, but the actual structures, the gross structure the assembly is quite different.
Yes.
Okay. Yeah, I wasn't so aware of that aspect of things, but that's quite important.
Well, why don't we move on? We're running short of time.
Right.
Okay. Yeah,
I mean, it's so fascinating that I could just listen to this all afternoon, but I'm sure everyone else is.
Awesome. Well, okay, we're wrapping up, I promise. But yes, so as Kurt and I, as our exchange just revealed, that metal valence has this really big effect on assembly anatomy for sure. And the next question that might sort of birth is, how would that alter the copper's electrochemistry, perhaps? And if you look at the voltammograms for copper 2 and copper 1 K16A, which I'm showing in panels A and B, they're quite similar at a first approximation, thermodynamically speaking. Their reduction potentials are both high at 700 millivolts, and the only distinction that you sort of get by eye is a current difference. And for copper 1 K16A, the voltammetric current is about half of its copper 2 cousin. And I'm predicting that diffusion is responsible for that. Copper 1's fibrils are larger and gelled, and consequently, its diffusion coefficient is smaller. And that was calculated out to 10 to the power of negative 15, which is 100 times less than copper 2 K16A, which was 10 to the minus 13. And this is where I want to bring quinones sort of back into the picture, shown over here. Unlike ascorbate, quinones reversibly reduce and oxidize. And we can track possibly how copper K16A electrocatalyzes this process, also using cyclic voltammetry. And perhaps copper 1 and copper 2 would interact with quinones differently owing to their different structures. And just a quick aside, I'm not using plastic quinone. Instead of that, I'm using the benzoquinone hydroquinone couple. And the reason for that is it's cheaper, it will actually dissolve in water, but it would still function identically to plastic quinone. And just to give us some common standing ground here, hydroquinone over here, this is electron rich and reduced, and it therefore oxidizes to form benzoquinone over here, which is oxidized and electron poor. Copper 1 K16A is electron rich, freshly prepared, and copper 2 by default is electron poor. All right. So if we look at the data across multiple scan rates, in the green plots, we have copper 1 K16A with either benzoquinone in A or hydroquinone in plot B. And in the red plots, we have copper 2 K16A also mixed with equivalent benzoquinone and hydroquinone. And across all samples, we can see that quinone addition ramps up the currents significantly. And that's reassuring because it tells me that the quinones are cycling relatively unimpeded by the assembly adsorptions to the electrode. More subtle, though, is how the quinones alter the wave shape, specifically this parameter called the peak current ratio. For reversible systems like copper 2 K16A, the ratio of the anodic and cathodic currents approach unity. And that means neither reduction nor oxidation is disproportionately favored. Chemically irreversible systems will have a peak current ratio that's either greater than or less than 1, meaning it's anodically or cathodically catalytic. So are the quinones actually altering the peak current ratios here? It's hard to tell that by eye, but I did the workup and I presented the ratios in the following bar graphs. The black and red bars correspond to the peak current ratio for copper 2 K16A and copper 2 K16A with hydroquinone. And these are all approximately 1 across several scan rates. And that means it's reversible. Adding benzoquinone, however, decreases the peak current ratio, meaning that the cathodic current is increasing relative to the anodic. And this contradicted my initial predictions because if you recall, hydroquinone is electron rich, copper 2 is electron poor. So I expected this HQ bar would go above 1 and BQ would not deviate from unity. So that's interesting, and it gets more interesting when you compare that to the copper 1 data, because first of all, on its own, copper 1 K16A appears to have some cathodic bias. Its ratio is at 0.5. And, again, strangely with the benzoquinone, that also does not seem to deviate from its control. But again, you look at hydroquinone, which is electron rich, and that gives me the result I expected for copper 2K16A, where it exceeds 1. And no, I did not swap up my sample labels. I double checked many times. Now, this confused me at first, but when I sat down and drew it out, I think it started to make more sense. Okay, in this first quadrant, I'm showing an electrode that is sweeping at negative potentials, and that means it's reducing. And adsorbed to it are some copper 2K16A assemblies. And this is what's happening. The assemblies are electron poor, so they're eager to take electrons from the electrode. Because of that charge hopping, these electrons can delocalize to the extremities, where they reduce benzoquinone to hydroquinone, which diffuses away thanks to entropics. That altogether causes IPC to increase. Now, when we sweep anodically over here, and that's to say the electrode is oxidizing, the adsorbed copper sites are now electron rich because the electrode just charged them all up. And we obtain our signal by reoxidizing the assemblies back to the apparatus. The hydroquinone does not appear to compete for these electrons, at least at a rate that's comparable to the electrode-driven process. Remember that peroxide experiment, substrate level reoxidation is slow. And that's also been recapitulated in the amyloid copper literature quite a bit, actually, in different sequences. So that's why I'm inferring anodic current isn't as affected or affected at all measurably. Now, the flip side, copper 1 K16A, the assemblies electrocatalyze hydroquinone in anodic conditions, and that drives current into the apparatus, raising IPA. But the assemblies, they're electron rich in cathodic conditions because the bulk was already largely reduced. And so I'm inferring it won't be as sensitive to hydroquinone reduction in this case. Okay, where did my mouse go? There we go. Oops, I went a little bit too far ahead. I just want to quickly say that, of course, I think more experiments are needed for this model, but I think that this is a solid start in understanding the mechanism for this Q-pool mediation that we have the seeds for. So I know that was probably intense and we've barely scratched the surface here, but if you've bought my argument that we've made a rechargeable electron sink, then it's time to consider what to do next with it. And I'm proposing that copper K16A could serve as a heterogeneous polymerization catalyst down the horizon. Our lab has shown that 6-amino-2-methaldehyde reversibly condenses to a polyimine on amyloid arrays. Polyimines, though, they're not so stable in water, but industrial polyamide and polyamine synthesis commonly involves redox fixation by an imine precursor through a copper redox catalyst. Copper K16A could satisfy both of these roles, I think. And if we demonstrate that copper K16A can fix an imine backbone into something water stable like an amide, then condensing aminoaldehydes would generate a polypeptide without a ribosome. And that would have large implications in the realm of alternative biochemistries, and it would be a step towards a synthetic metabolism mimic, which is not really precedented. So that's it for the data. I'd love to go on all day, but I'll just wrap up by referencing this cute metaphor that chemists like to use to describe the Ph.D. And that's the beloved reaction coordinate, and where we have, you know, a scientist catalyze a series of experiments that produces a degree. But I'm a peptide chemist, so I think the folding funnel describes the process more accurately. And the folding funnel has several mountains and valleys associated with it. And navigating these obstacles and barriers seems impossible at times, but the trick is really not doing it by yourself. And I don't know how to adequately or concisely relay my gratitude, but I'm going to start with my mentor, Dave. Dave, it's been quite the ride. I know you've always taught me that you can't learn anything without failing, and I have certainly learned a lot in my time here. And if I had to repeat graduate school 100 times, every single iteration, I do it with you, hands down. And I also have the best committee. I know every graduate student probably says that, but in my case, it's the truth. Cora, Vince, and Kurt, you have all given me countless insights regarding transition metals and peptides and unpaired electrons all over the years. And I also can't thank my committee without thanking Emily either. And Emily served on my committee for 3 years before moving to Penn State. And she was just so instrumental in my education. And of course, huge shout out to the Lynn Lab. It's been such a great place to work. I'm highlighting Young Sun and Lexi here because, like Dave said, COVID was a lovely interruption on several aspects of lab and personal life. And you both really helped me wrap up some final experiments that got destroyed by the shutdown. And I couldn't have wrapped this up without you. And of course, my collaborators, Anil, Katie, Andrew, Wei, Jason, and John, you were all the giants whose shoulders I stood on to see farther. So I'm just grateful for your continuing support throughout all of this.
Put some oomph in it.
Okay, so do I only twist?
Twist it, yeah, twist it. It's gonna come off.
Oh, it's moving.
See?
Okay,
There
ready?
you go.
Yeah.
Get you some oomph.
Okay.
Now just push it up with your.
Now push it up with your fingers down and it's at the end.
I love your tower stance. Yes. Tower. Get it, get it.